Hello, my name is Jewel Mitchell, and this is my exam mini video number 4 for Principles of Genetics 311. Today I'm going to talk about transcription in prokaryotes. A prokaryote is a single cellular organism that does not contain a nucleus. RNA is transcribed from a single-stranded binding protein. Here is an illustration of an RNA polymerase, which is represented by the orange circle. An RNA polymerase is an enzyme that produces the mRNA molecule. A prokaryote uses the same RNA polymerase to transcribe all of their genes. The polymerase is made up of all 5 subunits, which is called a holoenzyme. The DNA sequence where the proteins and enzymes bind to initiate the transcription process is called a promoter. The specific sequence of a promoter region is important because it determines how often the gene is transcribed. A consistent sequence comprises the most commonly encountered nucleotide found in a specific location of DNA or RNA. The start site of consistent sequence that is common to most bacterial promoters is the -35 region, and the promoter that is located 10 bases before the transcription start site is called the -10 box. The transcription start site is at position 1. There are 3 steps to the transcription process. First is initiation, where the creation of the first bonds between the RNTPs happen. Here we have the 2 strands where the shaded area represents the promoter region, and this eraser represents the RNA polymerase, with the sigma factor being the circle inside the RNA polymerase. The sigma factor associates with the core enzyme to form a holoenzyme. The next step is elongation, where the RNA polymerase undergoes a change in its shape and is no longer to bind to the consistent sequence in the promoter. The polymerase transcribes downstream and unwinds the DNA at the leading downstream edge, joining nucleotides and rewinding the DNA at the upstream edge. The orange string represents the coding strand, which has the same sequence as RNA. The blue string represents the template strand, where RNA is built on. Here the eraser is representing the RNA polymerase. The green string represents the terminator region. The final step is termination, where transcription terminates by inverted repeats caused by a hairpin. The formation of the termination hairpins destabilizes the DNA-RNA pairing, causing the RNA to separate from its DNA template. The presence of ribosomes moving along the mRNA in a 5'3" direction prevents backtracking of the RNA polymerase at the 3' end. Here the pink string represents the RNA being removed from the sequence, where the green string is the termination region. This eraser represents the RNA polymerase containing the Rho factor. The Rho factor has helicase activity, which is used to unwind the DNA-RNA hybrid, bringing it to an end. If the -10 box is deleted in a gene that codes for an enzyme to metabolize sugar, would cause an increase in the rate of transcription, which is called an up mutation, and this refers to any mutation in the promoter region that can trigger transcription initiation.